Hello everyone. Last time we have explained about the characteristics of the cell and the general organization of the cell together with the different type of cells such as prokaryotic cells or the eukaryotic cells together with their examples, that is a bacterial cell, plant cell or animal cell, and together with its various structures, that is presence of different type of cell organelles or the cellular components with respect to the plant cells, animal cells or the bacterial cells. Today we are going to explain the remaining part of the topic. So today here we have one table representing the definition of units for the measurement of the different cellular components or even the measurement or so as to measure the dimension or size of the different biological molecules or different structures or cell organelles or even the for the measurement of the cells or the tissues. So here is a table representing these units and its respective symbol and its conversion. So meter, the first one is a meter represented by letter small m and is equivalent to 1000 millimeter. So 1 meter is equivalent to 1000 millimeter. 1 centimeter is equivalent to 1 centimeter is equal to 10 millimeter into 10 raised to minus 2, 10 raised to minus 2 meter, or it is a 10 raised to minus 2 meter. So 1 centimeter is equal to 10 millimeter. 1 centimeter is equivalent to 10 millimeter, and it can be denoted as a 10 raised to minus 2 meter. Then 1 millimeter is equivalent to 1000 micrometer. 1 millimeter is equivalent to 1000 micrometer, or it can be denoted as a 1 into 10 raised to minus 3. 1 into 10 raised to minus 3 meter. Then micrometer is represented by micro, mu into small n, mu small m, micrometer, and is equivalent to 1000 nanometer, is equivalent to 1000 nanometer. 1 micrometer is equivalent to 1000 nanometer, or this can be denoted as 10 raised to minus 6 meter, 10 raised to minus 6 meter. Then 1 nanometer is equivalent to 1000 picometer. 1 nanometer is equivalent to 1000 picometer, and this can be denoted as 10 raised to minus 9 meter, 10 raised to minus 9 meter. Then angstrom unit represented by degree angstrom unit or capital A. Angstrom units, that is degree angstrom unit or degree capital A, and is equivalent to 100 picometer, is equivalent to 100 picometer. 1 angstrom unit is equivalent to 100 picometer and can be denoted as 10 raised to minus 10 meter, 10 raised to minus 10 meter. Then picometer, 1 picometer is equivalent to 1 picometer is equivalent to 10 raised to minus 12 meter. 1 picometer is equivalent to 10 raised to minus 12 meter. So this is just a table representing the different measurements or different units for the measurement of the various components or various particulate materials or various particles or the various molecules or the various structures whatever are present in the cell. So for the measurement of the different components of the cell, we can use these units. So this is just a table representing the measurements or the units of the measurement for the measurement of size of different molecules or different structures while the study of cells or its structures. Then this is another figure. This is again related with some biological components. The figure or the slide shows different biological components and having its a size or the measurement. So whatever the size, the approximate size of the certain biological compounds or the range of the measurements, the certain or definite range of the measurements or the size of certain components of the cells. So first one, in first small square you can see the atom. We know the atom. What is the definition of the atom? The atom is a part, a particle or a material. Atom is a smallest particle or a smallest structure. Atom is a smallest particle or a smallest uh, smallest part of the structure or a matter that will not break further. So this is the unbreakable. So it is the smallest unbreakable. It is the smallest unbreakable molecule or a structure. It is the smallest unbreakable part, smallest unbreakable part of the material or a matter called as atom. And the size of the atom is up to zero point 0.2 nanometer. So whatever the size of the atom, that is unbreakable part, that will not further broken down, that will not further broken down, that is unbreakable part, smallest part of the material, that is the atom. And the size of atom is up to 0.2 nanometer. Then another structure, that is a nucleotide. We know the nucleotide, that is a DNA, chain of nucleotide, DNA, deoxyribonucleic acid, DNA, the genetic material DNA, which is composed of a chain of nucleotides. The DNA is composed of DNA. We know the DNA or RNA having single strand or the double strand, and the single strand or the double strand having a chain of nucleotides. So in case of DNA, there are two chains of the nucleotides which are bound together in the form of DNA helix. So DNA is double stranded and RNA is single stranded. So the DNA or RNA which are having the chain of nucleotides, and the nucleotide that is consisting of the three components. Nucleotide is having three components, that is nitrogenous base, one of the nitrogenous base, then pentose sugar, and the phosphoric acid or the phosphate group. So these three components or the three structures or the components come together to form a nucleotide. So nucleotides are the components or the structures forming a either RNA or DNA, that is nucleic acids, that is a chain of nucleotides. So nucleotide is consisting of one of the nitrogenous base, out of five, that is purines or pyrimidines, that is nitrogenous base. Then pentose sugar, maybe ribose sugar or deoxyribose sugar, or third one, that is the phosphate group or the phosphoric acid. So these N, S and P, nitrogenous base, sugar and the phosphorus or the phosphate group, will form, these three components will form the nucleotide. So such a series of nucleotide or the chain of nucleotide will form the nucleic acids, single stranded RNA or double stranded DNA. So the size of the nucleotide is 1 nanometer. Size of the nucleotide is 1 nanometer. Then size of DNA helix, the size of the DNA helix is up to 10 nanometer. The size of the DNA helix is up to 10 nanometer. Then size of the chromatin material, whatever present in the nucleus. The size of the chromatin material is up to 100 nanometer. The size of the chromatin is up to 100 nanometer. Then the size of the nucleus present in the cell, size of the cell nucleus is up to 15 microns, is up to 15 microns. The size of the cell nucleus is up to 15 microns. Then size of the cell or the cell body width, size of the cell is ranging from 10 to 30 micron. The size of the cell ranging from 10 to 30 micron in a width. So this is the size of the cell. Size of the cell nucleus is up to 15 microns. Then size of the red blood cells, red blood corpuscles or the RBCs. The size of the red blood corpuscles is up to 6 micron. Red blood cells. We know the red blood cells, erythrocytes, which are without nucleus. The cells are without nucleus, having heme group in the hemoglobin. So red blood cells or red blood corpuscles. We know the red blood corpuscles are the erythrocytes, which are non-nucleated cells and which are containing the respiratory pigment, that is hemoglobin. So hemoglobin is a protein, a complex protein present in the RBCs and having heme group, and due to this heme group of the hemoglobin, the RBCs are having red color. So red color cells or red blood corpuscles which are red in color, and that's why the color of the blood is also red. So the color of the RBCs, red blood corpuscles, these are non-nucleated cells or without nucleus but containing hemoglobin as a respiratory pigment so as to carry or transport the respiratory gases, oxygen and carbon dioxide. So size of the RBCs is up to 6 microns. Then size of the arteriole lumen, the smallest blood capillary. Arterioles, we know the arteries and veins. We are well known to the arteries and veins which carry either oxygenated blood or deoxygenated blood. So one will form a part of arterial system, other vein, that is forming a part or the component of the venous system. So the lumen or the cavity present in the arteriole. Arteriole is the smallest blood capillary, which penetrates the tissues and supplying the nutrients or oxygen to the respective tissue or the cells. So arteriole, these are the smallest blood vessels. Arterioles or venules, these are the smallest blood vessels. So among the smallest blood vessels of the circulatory system, that is arteriole, that is the smallest blood vessel of the arterial system which penetrates the tissues and supplying blood to the respective cell or tissues so as to supply the nutrients or oxygen. So the lumen of the arteriole is up to 10 micron. Lumen of the arteriole is up to 10 micron. Then capillaries, which are somewhat larger in size, blood capillaries, having the size up to 8 to 10 micrometer. 8 to 10 micrometer. So thickness or the width of the blood capillaries, which are much larger in size, having the thickness up to 8 to 10 micrometer. Then the thickness of human hair is up to 100 micron. The thickness of the human hair is up to 100 micron. And the thickness of the blood vessels or the larger blood vessels which are somewhat larger or thicker, having more width or the width more than the capillaries, that is blood vessels, having the dimension or the width up to 1 centimeter. So these are the some of the measurements. These are the some of the measurements of the biological structures or various components either present inside of the cells or the outside of the cells. But these are the various structures or the cellular components either in the form of biomolecules or in the form of cells or in the form of tissues or the organs, but are having variable size or the thickness uh, with respect to their units or with respect to their measurements. So these are the some of the biological compounds or the materials which are either present in the cell or in the form of tissues or in the form of organs, but are having variable measurements or the size. So this is the slide just showing the measurements of the certain cells or the tissues of the living organisms. Then this slide showing the types of cells. So whatever the body of living organisms or the animals, particularly in the animals, these type of cells which are found among the animals. So there are different type of cells which are found in the animals and which are derived from the stem cells. So stem cell can be represented by the embryonic cells or the even the eggs or the fertilized eggs. We know there are the cells, that is the gamete cells, that is male gamete sperm or the female gamete egg. On fertilization will form a zygote in case of animals, and from this zygote the development of new one or the new organism will take place through the process of embryonic development. So actually this is the part of the developmental biology or the embryology where we can study the development of the new organisms of the particular species. And but in most of the organisms, wherever there is a sexual reproduction, wherever there are different, we know there are different modes of reproduction. We know there are different mode or the type of reproduction, that is asexual reproduction or the sexual reproduction. So in case of sexual reproduction, we can study or we have general idea about the process of reproduction, that is union or the fusion of male and female gamete, that is a sperm cell or the sex cell which are haploid, having haploid set of the chromosomes in the nucleus of the gametic cells, that is a sperm or eggs. So on the fusion or the union of the sex cells, that is male and female gametes, will form a zygote, and this zygote will then develop into a new organism through a process of uh, embryonic development, and embryonic development of the zygote starts with the cleavage. So the first division of the zygotic cell into two blastomeres, then successive mitotic divisions or the cleavages, then successive mitotic divisions or the cleavages of the zygotic cell will form a large number of cells and which are having similar shape size, which are having similar shape size and the composition. So whatever the cells or the cleavage cells formed during the embryonic development of the fertilized egg or the zygote, that is transformed into a large number of cleavage cells, and the number of cell get multiplied in the embryo. The number of cells get multiplied or increased in a number, and during each of the successive mitotic division, during each of the successive mitotic division or the cleavage, there is a doubling of the cells. So during each time, the number of the cells get doubled. So during cleavage, large number of cells with a similar composition or the equal composition with respect to its nucleus, then cytoplasm, or whatever the components of the cleavage cells or embryonic cells which are alike. All the cells produced, cleavage cells, during the cleavage are similar or the same type of cells, which will then transform into a stage of development, that is a morula. That is a ball of cells, morula, a solid ball of cells containing the cleavage cells. Then next phase of the development comes, that is a blastula, that is a single cell layer embryo called as a blastula. Then double cell layer embryo called as a gastrula. So there are, so, these are the steps. These are the steps uh, of the embryonic development. That is, fertilized egg or zygote will divide into two cells, then 4, then 8, then 16, then 32, then 64. Likewise, during each time there is a doubling of the cleavage cells or the blastomeres, and resulting into a formation of a solid ball of embryonic cells or the cleavage cells, or called as a morula, then blastula, then gastrula. So during the embryonic development, what happens? The number of cells get increased in the early phase of the embryonic development, and whatever the cells produced are having similar composition or the same type of cells or the cleavage cells. But as the development progresses, or as the embryonic development uh, goes forward so as to develop into new organism, there is a step, there is a step or a stage during which there is a development of three basic germ layers, that is ectoderm, mesoderm and the endoderm. Ectoderm, mesoderm and the endoderm. So these are three germ layers or the basic germ layers are formed in the development of the embryo. That is, embryonic development follows the blastulation, gastrulation, and during which there is a development or the formation of three basic germ layers, that is ectoderm, mesoderm and the endoderm. So up to this development of the embryo or during the development of new organism, there is a determination. There is a one stage, there is a one stage that happens during the embryonic development of the new organism called as the determination. So at this stage, that is when the gastrula forms ectoderm, mesoderm or the endoderm, three distinct layers of the cells of the embryo called as a determination. So at this stage there is a determination, and after which there is a differentiation. So when the embryo or the new organism develops from the fertilized egg or from the zygote, there are some steps or the stages during which there is advancement in the development of the embryo. At first there is a determination, means at the cellular level there is a sort of a decision or a sort of determination of the embryonic cells, which will then develop or get transformed into a particular type of tissues or the organs or the future parts of the embryo or the new organism. So that process is called as a differentiation. So embryonic cells when get determined, so this is a sort of predetermination or the determination so as to develop into a particular structure or the specific tissue or organ of the future embryo or the future organism. So that kind of activities or the events, successive events happening during the embryonic development is called as differentiation. So first there is a determination, and as per the determination, or there is a sort of determination of the base or the foundation for the future development of the new organism. So that is called as determination, and then followed by a differentiation. So differentiation of the new tissues or organs or the systems of the living organism are on the basis of the determination. So in this slide, we are going to explain different type of cells. So these are the different type of cells. In the center, there is a stem cell. In the center, there is a stem cell. So this stem cell is having the capacity or ability to differentiate or to develop into different type of cells or the different type of tissues which are responsible for the development of different tissues and organs or the systems so as to perform the particular biological function of the living organism, which is essential or necessary for the survival of the particular organism. So whatever the whatever the new organisms developed from the embryonic cells or the sex cells or the gametes, that is male and female gametes, as in case of animals, there is a fusion of gametes and formation of zygote, which will then develop into the new organism, as in case of animals. In case of plants, these are the seeds. In case of the plants, there are seeds. We know the seeds. So seeds are again derived from two sex cells, that is androecium or gynoecium, that is the sex cells or the gametes which are derived from the androecium or the gynoecium, that is gamete mother cells, and which will then develop into male part or the female part of the flowers, which will then fertilize and will produce the seed. So seeds will then germinate and develop into the particular type of plant. So this is a mode of sexual reproduction in a plants. So similarly, among the animals, the sexual reproduction will occur with the help of the sex cells, that is male gamete and the female gamete, as the cells shown in this slide, that is a sex cell, that is sperm and the egg. So on the fusion of the male and female gamete, there is a formation of zygote or the animal seed. We can say it as a animal seed. So from the animal seed, the animal of the particular species will develop. So in this slide, we see the stem cell in a center. So stem cells are the cells called as the totipotent cells. Stem cells are the totipotent cells. So totipotency means these cells are having ability or the capacity of development of all the type of cells, all the type of tissues of the particular organism. So in the center, you just see a stem cell. So stem cell nowadays or in a modern days, the stem cells are used for the therapeutic use, for the purpose of treatment, therapeutic use or the treatment. So for the purpose of treatment of particular disease or disorder, for the treatment of particular disease or disorder. So this is a modern concept, or this is the new approach of the treatment of the certain disease or the disorders in the human being, that is use of stem cells for the therapeutic purpose or the for the treatment purpose. So we can use the stem cells so as to replace the particular organ or the particular tissue by new one. So use this is just a idea or the modern approach of the treatment or the therapy, that is the use of stem cells. So basically the stem cells are the totipotent cells and are having ability or capacity to differentiate or to develop into almost all type of cells of the particular species or the organism. So in the center there is a stem cell. So from the stem cell we can uh, replace or we can have the different type of cells on its differentiation. So this can differentiate into different type of cells, and thus This is the new approach of using stem cells for the treatment of the different abnormalities or the diseases, so that we can have the particular structure while using the stem cells. So stem cells can be differentiated into muscle cell, then fat cell, bone cell, blood cell, then nerve cell, then epithelial cell, immune cell, or even the sex cell also. So the stem cells, these are these may be embryonic cells. These may be the embryonic cells, or there are certain structures. There are certain structures or the organs present in the body which can be a source, of the stem cells. So this is the slide showing the types of cells. We know the muscle cells. So muscle cells are the elongated cells representing different type of muscles. That is smooth muscle or skeletal muscles or the fibrous muscles or the cardiac muscles. So there are different type of muscle cells or the muscles which will overall form a body musculature among the animals. Then another type, that is fat cell. We know the fat cell. So these are the cells which are mainly responsible for the deposition or the storage of the fats or the adipose tissues that will form the adipose tissue. So which are mainly containing lipids or the fats as a reserve food material in the fat cells. Then bone cells. So bone cells again will form a structural unit of the endoskeleton. So whatever the bones present in the body of vertebrates representing the endoskeleton, providing mechanical support to the body, which are derived from the bone cells. Then blood cell, that is uh, either RBCs, WBCs. So here these are the blood cells representing the nucleated cells, means these are the white blood corpuscles or the WBCs. So the cells which are having nucleus, that is the blood cells which are with a nucleus representing white blood corpuscles. So these are the blood cells. Then stem cells can also be differentiated into the nerve cells or the neurons. Nerve cells, we are well known to the nervous tissue or the neurons or the nerve fibers or the nerve cells which are uh, responsible for the transmission of the impulses or the sensory stimuli or the impulses from one side or one end to the other end of the body. These are the longest. These are the longest cells. So nerve cells are the longest cells among the all type of cells of the living organism. Then another type, that is epithelial cells. EP means upon. EP means upon. So nerve cells are responsible for the conduction of the stimuli or the impulses, that is neurotransmission or the conducting impulses, either sensory or the motor. So motor impulses or the motor impulse means a response, and sensory impulse means a sensory stimuli. So this can be transmitted through the nerve fibers or the neurons or the nerve cells. Then another type of cells, that is epithelial cells. EP means upon. So whatever the surface of the tissues or the living organisms or the organs consisting of epithelial cells. So epithelial tissue or the epithelial cells may be simple or may be compound or the stratified epithelial tissues can occur in the body in the different regions, epithelial cells. Then immune cells, these are again the blood cells representing the lymphocytes. These are the nucleated cells called as lymphocytes, and which are responsible for the development of the immunity, that is synthesis of antibody. Antibody synthesis will happen with the help of lymphocytes or the immune cells. So this is the chart representing the different type of cells which are useful or which are responsible for the formation of formation or the development of different tissues and organs or the systems of the living organism so as to perform different biological functions of the living organism so as to survive or so as to grow, so growth, development, then reproduction. So whatever the life activities or the life processes performed by the living organism that are only due to the different type of these cells which compose it to form the tissues or the organs or the systems of the living organism and will perform the different biological functions during the life processes or during the survival or during the lifespan of the living organisms, right from the development, that is growth, then complete development up to the end of the age of the particular organism. So these are the different type of cells which can be derived from the stem cells. So this is again another slide representing the some sort of a plan or a chart. This is the diagrammatic representation. This is a chart representing the representing the plan or the diagrammatic representation of the development of the new organism or the new cells. So first, the yellow color cell will explain all these steps one by one. So first cell, that is a totipotent embryonic stem cell. Totipotent embryonic stem cell. So totipotent means again the cell having a capacity of differentiation of the single type of cell into all or the almost all type of cells of the living organism are derived from the totipotent cell. So totipotent cells can be the embryonic cells. So these are the embryo. So even the zygote or the cleavage cells or the embryonic cells of the embryo can also act as or serve as a totipotent cell. So totipotent cell and the stem cells. Stem cells are somehow different. Stem cells are somehow different from the embryonic totipotent cells. Totipotent embryonic cells or the totipotent embryonic stem cells. So stem cells or the embryonic cells or the totipotent cells which are having ability or capacity of its differentiation into different type of cells and tissues which are responsible or which are essential for the development of the new organism. So the first cell representing totipotent embryonic stem cell. So this totipotent embryonic stem cell will be differentiated further into the pluripotent embryonic stem cell. Pluripotent, plural. So single cell, many cells. Pluri means several cells. So pluripotent embryonic stem cell. So totipotent embryonic stem cell will then develop or transform into the pluripotent embryonic cells. And this pluripotent, pluri means many, pluri or the plural, singular, plural. So these are the just terms, singular, single, plural, many. So the pluripotent embryonic cells. So the totipotent embryonic stem cell, totipotent embryonic stem cell or the cleavage cells or the embryonic cells, whatever the number produced from the zygotic cell. So whatever the number of cells present in the embryo in the form of blastula or gastrula consisting of single layer or double layer or triple layer, and that will be transformed into a pluripotent embryonic stem cells and which will then develop. So single totipotent embryonic stem cell will be transformed into three type of pluripotent embryonic stem cells as I have mentioned uh, earlier, that is formation of three basic germ layers or the development of three basic germ layers representing ectoderm, mesoderm or endoderm. Ectoderm, mesoderm or the endoderm. So these ectoderm, mesoderm or the endoderms, these are three basic germ layers. Germ means germination or the development or the formation. So three basic germ layers which are derived from the embryonic cells or the cleavage cells, which are no doubt are totipotent. So these are the totipotent embryonic cells, or can be termed as or explained or described as a stem cells also. But the stem cell is somewhat new approach or the modern term used for the cells having ability or capacity of its differentiation into almost all type of cells of the particular organism, and which can be used for the purpose of treatment or the therapeutic use. So stem cell is new approach, but totipotent embryonic stem cell, that is the embryonic cells which are having ability of its differentiation into different type of cells or the all type of cells of the living organism. So the first cell, that is totipotent embryonic cell or the stem cell, that will form, that will develop or get transformed into the pluripotent embryonic stem cell. Pluripotent embryonic stem cell. Stem means base. Stem means a trunk or the important part of the plant representing the stem. So that is the main part of the trunk as in case of plants. So stem. So this is the important part. So the basal or the important part of the development of the embryonic development, and thus the term used to be as a stem cell. So totipotent embryonic stem cell or a zygote, which will then develop into the pluripotent embryonic cells, as in case of embryo, that is blastula or gastrula, possessing many number of the cleavage cells, and that are pluripotent embryonic cells. So whatever the cells developed or formed during the proliferation or the multiplication or the successive stages of the cleavages of the zygote, resulting into formation of large number of cleavage cells, that are pluripotent embryonic cells. So which are then transformed into the ectoderm, endoderm or mesoderm. Endoderm, mesoderm or the ectoderm. So these are the three basic germ layers, means these are cells, these are the embryonic cells or the cleavage cells of the embryo, that is blastula and gastrula. So some of the cells of the blastula and gastrula are transformed into the pluripotent embryonic cell in the form of three basic germ layers or three type of cells, that is pluripotent embryonic cells such as ectodermal cells, mesodermal cells or the endodermal cells, and this can be represented in the figure as a ectoderm line, mesoderm line or the endoderm line. So these are the pluripotent embryonic stem cells, that is which are developed during gastrulation. So these are named or these are determined, as a ectoderm, mesoderm and endoderm. So whatever the tissues develop further, whatever the tissues or the cells develop further during the embryonic development, that is after gastrulation, that is organogenesis, or the formation of organ rudiments, that is a base or the foundation for the differentiation of the different type of tissues and organs. So just after the gastrulation, when there are three basic germ layers are formed representing ectoderm line, mesoderm line or the endoderm line, which then again get transformed into the multipotent stem cells. So pluripotent embryonic stem cells, that is basic germ layers, ecto, meso and endoderm, will be transformed into the cells, which are again simply redividing and forming more cells or addition of cells due to the multiplication, continuous process of the multiplication of the embryonic cells, that is transformation of the ecto, meso and endodermal line into multipotent stem cells. Multipotent stem cells means again each of the cell of ecto, meso or endodermal pluripotent embryonic cells will divide and transform into a multipotent stem cells. Multipotent stem cells, and these multipotent stem cells will again be transformed into the respective type of cells or the tissues, which are then forming a component or the composition of the organs or the systems, and the development of the overall the entire body of the living organisms, which has been composed of different systems. So in this figure you can see the multipotent stem cells will be transformed into the different type of tissues, different type of tissues or organs. So we can explain them one by one. So the ectodermal line will be transformed into the multipotent stem cells for the organs or tissues which are of ectodermal origin, and the example of which are the skin and neuron. Skin and neuron. So entire body wall or the body covering or the skin is ectodermal origin. Even the nervous system is also ectodermal in origin. Nervous system. So in a chart, in the base you see the gray colored cells, that is ectodermal line, or the multipotent stem cells, gray colored cells, which are then transformed into a skin and the neuron or the nervous tissue or the nervous system. So skin or the body wall or the entire body covering, so that is ectodermal origin, that is ectodermal in origin, that is skin and neuron. In a central red color cells, that is mesodermal line, will transform into multipotent stem cells, which are then transformed or differentiate into the heart muscles, that is cardiac muscles, and the red blood cells. So these are the components mesodermal in origin. It means the circulatory system. It means circulatory system, that is blood vascular system, that is arterial system, venous system, lymphatic system, their vessels, even heart, they are even blood is derived from the mesoderm. So mesodermal origin. So visceral organs, many visceral organs, or the even musculature, body musculature, or the broadly we can say the organs or tissues or the various structures of the body which are usually present in the middle portion or the middle part or in middle of the body are of mesodermal origin. So mesodermal line will transform into a multipotent stem cells, and these multipotent stem cells will be transformed into the heart muscles and the red blood cells, which are mesodermal in origin. It means circulatory system or blood vascular system is of mesodermal origin. Then endoderm line, that is yellowish cell, endodermal line, that is endoderm, cell line, that is having endoderm cells which are transformed into a multipotent stem cells of the endoderm, and which are then differentiated into lung and pancreas. Lung and pancreas. So lung, respiratory organ, pancreas is again a uh, a duplex gland, associated digestive gland, accompanied with the digestive system. So the structures, that is which are or the organs or the tissues or the structures whatever present in the central portion or at the interior of the body of organisms is derived from endoderm line or the endodermal in origin. So digestive system, then respiratory system, digestive system or the respiratory system, these are endodermal in origin. So this is how the development progresses from the totipotent embryonic stem cells to the pluripotent embryonic stem cell, then to the multipotent stem cells, and which will then in a final phase of development or at the end of the development of new organism, that is when the new organism completely developed into a new organism. So when the development completed, when there is a complete development of the new organism at the end of development, will result into well defined or well developed different type of tissues and organs or the systems so as to perform the particular biological function of the living organism, that is either neurotransmission or the protection of the body, neurotransmission by nervous system, that is control and coordination of different activities, that is sensation, then sensation, that is receiving sensory stimuli from the surrounding and supplying them or sending them to the central nervous system and again performing a particular action in the form of response. So the function of nervous system or the function of the skin, that is protection of the body, receiving various sensations through the skin, that is uh, the function of the uh, different system, that is the system which are derived from the ectoderm, that is skin or the nervous system. Then mesodermal structures which are then develop into the circulatory system or the body musculature, which is responsible for the movement of the body or the circulation of the blood through the circulatory system so as to supply nutrients to the cells or so as to carry the respiratory, transport the respiratory gases O2, CO2, or so as to collect or absorb the nitrogenous waste or the carbon dioxide from the tissues or cells and to carry them towards the excretory organ, that is the system, that is excretory system, then circulatory system will be developed from the mesodermal line or the mesoderm. Then endodermal system or organs, that is digestive system or respiratory system or the reproductive system, which are functioning properly on the complete development or after the complete development. So this is just a pattern of determination and the differentiation of the different cells. So how the development of new organism will occur. So this is just a pattern or the diagrammatic representation of the transformation of the cells, embryonic cell to the pluripotent or multipotent cells, and at the end of the development there is a complete development of the different tissues and organs or the systems of the living organism to perform different biological functions, maybe protection, sensation, then nutrition, respirations, then circulation, excretion, or neurotransmission. So whatever the biological functions of the living organism that are performed by the different systems after the complete development or the cellular transformations at the end of the development. So the chart shows on the right side you can see the adult bone marrow, skin, cord blood, or deciduous teeth. So these are the some structures which are having either multipotent or the pluripotent stem cells and which are having capacity to get transformed into the human embryonic stem cells or the induced pluripotent stem cells. Means there are certain organs of the tissues which are having ability to induce or to stimulate certain cells to get transformed into the stem cells. As in a liver, for example, in a liver, spleen, bone marrow, liver, spleen, bone marrow, these are the some organs which are having certain type of cells or the regenerative type of cells or the totipotent cells which can be transformed into the different type of cells. So this is just a chart representing the type of cells, that is stem cells or the embryonic cells useful which are useful for the development of new organisms. So this is just a plan or the cellular components of the cells or the chart representing the diagrammatic representation of the development of new organism or the performance of the new organisms while their development, growth or survival of the life. Then last point, that is the characteristics of the living system. Characteristics of the living system. So these are series of the characteristics of the living system. We'll explain them one by one. Organized. Organized. So this is the organization, that is the uh, organization, that is the formation of different cells, tissues, organs, systems, and finally the development of new organism. So organized, that is organisms are organized. Organisms are organized. Organisms are organized with a sequential events. With a sequential events since the development or the birth up to its death. So whatever the activities from birth to death, whatever the activities occurring in the life of the living organism, that is right from birth up to the death, even prior to birth there is a development. Even there is a developmental phase, there is a developmental phase that is also a part of the living system. So prior to the birth of the living organism, there is a development. So from development of the egg, or as we have explained in a previous slide, that is a development of the new organism through the successive divisions or the successive transformation in the embryo, and will develop into a new organism, or the embryo into a new organism, or the young stage which take the birth, young stage which take birth or which hatch from the egg, or which emerge out of the egg, or which take the birth after the complete development, as in case of viviparous animals. So organized, the single word organized represents the characteristics of the important characteristic of the living system, that is organisms are organized. And while organization of the organisms, there are several steps which can be explained as a simple words which are given in the bracket, that is a cell, tissue, organs and organ system. So organ system, so cells will form the tissue, tissues will form the organs, organs will form the organ system, and organ systems will form organism. So this is just a organization. This is just a organization of the organism will pass through the several steps, that is the cell right from a zygote or the embryo, then tissue, then organs, then organ system, and finally will develop into a new organism, that is organized. So this is the important characteristics of the living systems, that is organized, that is organization. So organisms are organized, but this organization of the organisms pass through the cell, tissue, organs and organ system. Then made up of cells, living system made up of cell. Organisms are made up of cells. Metabolism, organisms get materials and process them for energy. So whatever the nutrients, whatever the whatever the nutrition or the nutritional components in the form of food containing different foodstuffs, may be proteins, fats, carbohydrates, minerals, salts, water, all of which are essential for the metabolism. So organisms get these materials in the form of nutritional components or the food, and are then processed in the cell, are then processed in the cell, may be digested in the digestive system, then absorbed into the blood, then sent or supplied to the cells, then reach into the cytoplasm where they are processed, actually used or assimilated, that is assimilation, actually used in the cells cytoplasm for the requirements of the cells or the tissues or the body of the organism, that is process. So organism getting these materials either in the form of nutrients or water or respiratory gas, oxygen, and which are then processed in the cell for the energy purpose. Even for the building or the formation of the cell itself, whatever the components of the cells that are also derived from the materials which are brought into the cell or which are supplied to the cell. So the metabolism, organisms getting materials and processing them in the cells, using them in the cells for the synthesis of protoplasm or cytoplasm or the various components of the cells, for the growth, for the process of mitosis, for the process of repair, for the supply of energy so as to perform different biological functions. So whatever the functions performed by the body needs energy for the movement. For example, there needs the energy, so this energy will be supplied by the cell itself through the process, that is metabolism. So metabolism is also one of the important characteristic of the living system or a living organism, that is metabolism. Organisms get material and process them in the cytoplasm of the cells where it is transformed into energy. Then homeostasis. Homeostasis is also the important characteristic of the living system. Homeostasis, that is organisms, maybe plants, animals or the microorganisms, they themselves regulate their internal environment. So as to maintain the normal internal environment, there are some sort of mechanisms which are also happening in the cells or in the tissues or organs themselves so as to maintain a normal internal environment or the physiological condition called as homeostasis. That is maintenance of the water contents, maintenance of the electrolyte, maintenance of the sugar. So whatever the components present inside of the cell that are maintained at their optimum level, that is called as homeostasis. So organisms regulate their internal environment themselves with the help of cellular functions or the cellular activities, homeostasis. Then growth. Organisms have the capacity to grow and develop. So with the help of the metabolic activities, the animals are capable of their development or their growth or their survival, even reproduction. Organisms have the ability to reproduce. So organisms, they can propagate, they can continue, they can reproduce the individuals of the same species by themselves. So this is also the ability or the important characteristics of the living system or the organisms to undergo growth, to undergo reproduction. So organisms are capable of reproduction. So they can reproduce, they can produce the organisms similar to them and having this ability of reproduction, that is propagation, multiplication, increasing their number. So this can also be one important characteristic of the living system, that is reproduction. So there are various modes of reproduction, sexual, asexual mode or even parthenogenesis. There are different modes of reproduction which are seen or observed in a different species of the organisms. So organisms have the ability to reproduce. So they undergo the reproduction. Then stimuli. Organisms respond to stimuli. Organisms respond to stimuli. So whatever the external stimuli or the internal stimuli that are received by the receptors or the sensory cells or the sensory organs. So there are well developed system so as to receive the sensory stimuli, external stimuli from the external environment or the surrounding environment, that is eyes, ears, nose, tongue, skin. These are the sense organs which are receiving several sensory stimuli from the surrounding. So organisms receiving various stimuli from the surrounding and even they respond them, means there is a sort of response, there is a particular action or a kind of reaction against the stimulus, so as to uh, uh, coordinate the different activities of the living organism. So various type of senses or the stimuli which are received by the different sense organs and messages of which are sent to the central nervous system, and again central nervous system will pass another message or the impulse to the effectors, that is muscles or the glands, so as to so as to react against the particular stimulus, so as to show the particular action, so as to perform particular activity against the stimulus, so as to react, so as to control and coordinate its body activities. There are different stimuli which are received by sense organs, and even the respond or the in the form of action against the stimulus is also given by the living organisms or the living systems. So stimuli, organisms respond to stimuli. Then adaptations, that is organisms adapt and to change, adapt and change. Organisms adapt to change means whatever the surrounding conditions, either internal environment or the external environment, which influences the life activities. So animals or the living systems themselves get adapted, get modified themselves so as to resist, so as to withstand, or so as to tolerate the adverse conditions of the external environment or the internal environment, and get changed so as to tolerate, so as to resist. So this will lead to the evolution or the variations. This leads to the variations or the changes in the body of living organisms. So living systems, they are having important characteristics, that is adaptations or the modifications in them as per the changes taking place in the surrounding environment or in the internal environment or in the body. So whatever the internal or external changes taking place in the external environment or in the internal physiological conditions, the organisms themselves get adapted, get modified and get changed so as to resist, so as to withstand the different adverse conditions of the internal or external environment. This is called as adaptations. Then, so this is about the characteristics of the living systems. Then these are the some of the branches of the cell biology and other biological sciences, or the wherever the cell biology is concerned, where the cell biology is related with another biological sciences, that is cytotaxonomy, that is cytology and taxonomy, cytogenetics, cytology and genetics, cell physiology, cytology and physiology, cytochemistry, cytology and chemistry, ultrastructure and molecular biology, cytopathology, cytology and pathology, cytoecology, cytology and ecology. So these are the different branches of the biological sciences. These are the different branches of the biological sciences wherever the cell biology or the cytology is related closely. So this is what about the topic first. So today we'll stop here. Next time we'll continue with next topic. Till then, bye. Thank you.